Let's continue with Earth's spheres, yes? We learned about 4 spheres, the atmosphere, biosphere, geosphere, and hydrosphere. In our previous videos, we saw about atmosphere, geosphere, and hydrosphere. Now let's see in detail about biosphere. Biosphere. How is biosphere different from all the other spheres we have learnt? Differences: they are made up of cells, which make them living organisms. There are 5 different kingdoms in biosphere we know of: animals, plants, fungi, protists, and monera. Cells. The cells are the smallest structural and functional unit of an organism. Definition: a cell is defined as the smallest basic unit of life that is responsible for all of the life's processes. You're supposed to memorize this definition. A cell is defined as the smallest basic unit of life that is responsible for all of the life's processes. A cell can replicate itself independently. Hence, they are known as building block of life. You can see that they can replicate by itself. Robert Hooke was the first biologist who discovered cells in 1665. Moving on to the next slide. Here we are going to see a timeline of how technology and science advanced. In the year 1663, Robert Hooke, a scientist, he studied bark and fossils with microscope, and he came to understand that everything is made up of cells. And Robert Hooke was the person who coined the term cells. And then in 1674, Anton van Leeuwenhoek observes living organisms under a microscope. What he basically did is, he said that the cells are living, whereas Robert Hooke thought they were not living. Next, the scientist Matthias Schleiden concludes that all the plants are made up of cells. Then in 1839, Theodor Schwann reaches the conclusion that all the animal cells are also made up of cells. In 1855, Rudolf Virchow proposes that cells are only made up of other cells. That means that a cell can only make another cell. Then in 1930, scientists developed several types of electron microscopes that are 5,000 times powerful than what was initially discovered. That's the light microscope. So let's see a little bit about microscopes. The cell theory could not have been developed without microscope. Yes, the microscope we use today have the same function as those that were used 200 years ago. This was the one that was first discovered, the light microscope. And this one is the modern electron microscope. See the different parts of this light microscope. Starting with the source. This is the light source. This is where the light comes from. The light source illuminates the specimen by shining bright light through it. So you place a specimen here and it illuminates the specimen by shining bright light through it. Next part is the diaphragm that you see in here. This is a rotating dial that controls the passage of light through the stage. Numbers on the dial indicate the relative amount of the light passing, with 5 being the most and 1 being the least. So this is the fine focus knob used to make small focus adjustments, especially when using high power objective. And this is a coarse focus knob. Moves the stage up and down quickly. Used to find a specimen when using low power objective. Next is the stage. This is the stage. It holds the slide and contains an opening that allows the light to pass through the specimen on its way to the objective. That's the objective. These are the objective lenses. Objective lenses. Objectives produce most of the magnification. The higher power lens, the blue striped ones, magnifies about 40 times. And the low power lens, the yellow stripe, magnifies about 10 times. The arm. It supports the lens, the mirror, and the body tube. The microscope should be carried with one hand holding the arm and the other hand under the base. That's how you should carry it. One hand you'll be holding it in the arm and the other hand you'll be placing it below the base. Rotating head. This contains mirrors and allows the body tube to rotate 365 degrees. This can be rotated 365 degrees. That's the body tube. The light passes through it from the head to the eyepiece. From here to the eyepiece. This is the eyepiece. So what does the body tube do? It passes the light from the head to the eyepiece. The eyepiece is where one views the enlarged object. It contains a lens called the ocular that further magnifies the specimen by 10 times. So here there's a lens that magnifies by 10 times. So what's happening? Here the the magnification can be 40 times with the blue striped one and 10 times with the yellow striped one, and then it further magnifies here by 10 times. So those are the parts of the microscope. Moving on to the next slide, we have cells. Cells can be divided into two: prokaryotic, which are single cells, and eukaryotic, which are mostly multiple cells. That means it can sometimes also be a single cell, but whereas the prokaryotic, it can only be single cell. The prokaryotic has got monera, which are the bacteria. So if you can see in here, that's just one single cell, and it has an oral groove and an anal pore. So what it does is the food intake happens through the oral groove, and then the food goes in and it forms a vacuole, and then the vacuole goes down, it gets digested and nutrients get absorbed, and then the waste is released through the anal pore. So it's a simple mechanism, just a single cell doing all of it. All the functions are performed by just one single cell. What were all the functions that we saw? Taking in the food, processing the food, digestion of the food, absorption of the food, and then releasing of the waste. Next, we'll see about eukaryotic cells. In eukaryotic, it's divided into plants, animals, fungi, and protists. We'll see in detail about protists. Example for our protist would be euglena. So this is also a kind of one-cell organism. That's what we saw. Eukaryotic are mostly multiple cells, but sometimes it can be one single cell. So the protist is almost like the monera, just that it has some organelles in it, whereas in the monera you did not see any organelles in it. Bacteria are all unicellular, whereas protists, they can be unicellular, some can be multicellular. All prokaryotic has no nucleus or membrane-bound organelles. All eukaryotic have a nucleus and membrane-bound organelles. Moving on to the next slide. Next is the nucleoid. So the bacteria, they don't have nucleus to hold their nuclear DNA. Instead, they have something called nucleoid, wherein the DNA is found. This structure has no protective membrane, but it's tightly packed DNA and RNA molecules in it. Next is the plasmid. What's plasmid? In addition to nucleoid, bacteria have plasmid. Plasmids are small DNA molecules that can hold extra genes that may be used when cell experiences certain conditions. These little piles of DNA can also be exchanged between bacterial cells. Next, bacterial villi. Long thread-like structures coming off of the cell's surface. Bacteria can use these to bind to other bacterial cells to exchange genetic material. So now that we have learned about the bacteria, which are the unicellular prokaryotic cells, let's move on to the eukaryotic cells. That's the multicellular cells. The first one was the protists. Yes, many protists acts as pathogen to humans. Example, the disease malaria is caused by the protist Plasmodium falciparum. What are the different modes of transportation that these protists use? The paramecium use cilia. Cilia are thin, very small tail-like projections that extend outwards from the cell's body. Cilia beat back and forth, moving the protist along. Next, amoeba. They use something called pseudopods. The word pseudopod comes from the Greek word meaning false feet. If an amoeba is cut in half, the half with the nucleus will survive while the other half will die. Next, euglena. It has something called flagellum. Like the bacteria that we saw previously, even the euglena has flagellum. Flagellated protists are those protists with tail-like projections called flagellum, which the organism whips in a circular motion to propel itself forward. Next, so that we have learned about one of the eukaryotic cells, the protist, let's move on to the plants and animal cells. Animals are multicellular organisms that form the biological kingdom Animalia, and plants are also multicellular organisms of the kingdom Plantae. Let's see how they are different. Animals are heterotrophs, that's they cannot produce its own food. Instead, they take nutrition from other sources. Whereas plants, they are autotrophs. They can produce their own food, water, and carbon dioxide. Animals consume oxygen. Plants, they both consume and produce oxygen. Animals are able to move. Plants generally don't move. Animals reproduce sexually, whereas plants, they reproduce sexually or asexually. What are the key similarities between the animal cells versus the plant cells? Animal cells and plant cells are both eukaryotic cells, yes? Meaning they both possess a true nucleus. So as you can see in this picture, they both have a nucleus. DNA is enclosed in a membrane-bound nucleus, so it's bound by some membrane. That's the similarity between both. They have a membrane bound about the nucleus. Both animal and plant cells are multicellular organisms. We know that. So they can multiply by themselves. And also they have several membrane-bound organelles. Not just the nucleus, they have other kinds of organelles which are also membrane-bound. Moving on to the next slide. What are the major differences in animals and plant cells? We know that they have nucleus, that's the similarities. They have all these organelles which are also membrane bound. So all these organelles are similar. But then the few extra organelles present in plant cells which make them different from the animal cells. What are those? They have this big vacuole, and they have cell wall, and then they have chloroplast. These are the three organelles that are only present in plant cells. You will not see these organelles in the animal cells. What are those again? A big vacuole, a cell wall. Please do not confuse this with the cell membrane. That's different. It's a thin covering that covers the entire cell, which is present both in animal cell as well as in plant cell. If you can see in here, cell wall is just present in plant cells, not in animal cells. So what are the three things that are present only in plant cells? Vacuole, cell wall, and chloroplast. So if you have understood that, let's move on to the next slide. Cell structure. The cell structure comprises individual components with specific function essential to carry out life's processes. Now let's learn about each of these organelles in detail. Let's start with cell wall and plasmodesmata. Remember, I just told you that cell wall is only present in which cell? The plants. The animal cells do not have cell wall. So now we are learning about plant cell in this case. In addition to cell membrane, plants have cell wall. Cell walls provide protection and support for the plants. In land plants, the cell wall is mostly made up of cellulose. Unlike cell membranes, materials cannot get through cell walls. This would be a problem for plant cells if not for the special opening called plasmodesmata. Yes, there has to be some movement between the cells. But then they are saying that the cell wall, it does not allow any materials to get through. So that's where these small openings called plasmodesmata help in exchange of materials in and out of the cell. Can you see that? Small openings called plasmodesmata. They help with the movement of materials in and out of the cell. Okay? These openings are used to communicate and transport materials between plant cells because the cell membrane are able to touch and therefore exchange needed materials. What it means is two cells that are close together are almost close. If there is another cell in here, they are close nearby. So there is transfer of material from this plasmodata to this plasmodata. So there is exchange of material in between two cells. Next, let's move on to plasma membrane. Plasma membrane is found both in animal cells and plant cells. Plasma membrane is the one that encloses the cell, okay? Plasma membrane or cell membrane. You can also say it as cell membrane. The membrane enclosing a cell is made up of two lipid layers called bilipid layer. Can you see that there are two lipids, one in here and one in here? Two lipid layers called bilipid membrane. The lipids that present in the plasma membrane are called phospholipids. So the phospholipids have different parts. They have the head, that's this one, and then they have the tail. The head is hydrophilic, which means water loving, hydro means water, and philic means loving. And then there is this tail, which is water hating, hydrophobic. Hydro means water, philic means loving. Hydro means water, phobic means hating. So let me tell you again. Head is hydrophilic, water loving, and tail is hydrophobic, water hating. Moving on to the next, channels and pores. These are present both in animals and plant cells. A channel is found in the cell's plasma membrane. This channel is made up of certain proteins that control the movement of molecules, including food, water, in and out of the cell. This is what helps the movement of particles like food and water in and out of the cell. Okay. Next, moving on to cell wall septum and pores. This is mostly found in the fungal cells. Remember, in the 5 animal kingdom, we saw that eukaryotic has animal cells, plant cells, and also fungal. So these cell wall septum and pores are found in fungal cells. The fungal cells have both cell membrane, cell wall, like the plant cells. They are more kind of like plant cells. They have cell walls as well. A cell wall provides protection and support. Fungal cell wall are largely made up of chitin, which is the same substance in insects' exoskeleton. Because materials cannot get through cell wall, fungal cells have special openings called pores. Remember, there was something similar like the pores in the plant cells. What was it? The plasmodesmata. The plasmodesmata, remember? So it's the same concept in the fungal cells. The pores have the same concept as plasmodesmata. Some fungal cells also have septum, that are special internal walls between cells that are found in long tube-shaped strings or strands called hyphae. If you can see in here, this would be the septum. Septum is the one that's in between two cells. Let me read it again. Some fungal cells also have septum, that are special internal walls between cells that are found in long tube-shaped strings or strands called hyphae. The nucleus is the control center of the cell. It's the largest organelle in the cell and it contains the DNA of the cell. As you can see here, that's the DNA. DNA and nucleolus. DNA, which is the deoxyribonucleic acid, contains all the information for the cells to live, perform their functions and reproduce. Inside the nucleus is another organelle called the nucleolus. The nucleolus is responsible for making ribosomes. The circles on the surface of the nucleus are called nuclear pores. These are where the ribosomes and other material move in and out of the nucleus. Ribosomes. Organelles that help in synthesis of proteins are called ribosomes. Ribosomes. Organelles that help in synthesis of proteins are called ribosomes. With a specific information unit called messenger RNA, these make proteins. Some ribosomes are found in the cytoplasm, but most are attached to the endoplasmic reticulum. While attached to the endoplasmic reticulum, ribosomes makes protein that the cell needs. So that's where the ribosomes are attached. If these are the endoplasmic reticulum, those are the ribosomes. Next, endoplasmic reticulum. It's a network of membranes throughout the cytoplasm of the cell. When ribosomes are attached to it, it's called rough endoplasmic reticulum. And smooth endoplasmic reticulum are when there are no ribosomes attached to them. So those are the smooth endoplasmic reticulum. And those are the rough endoplasmic reticulum. Can you see the ribosomes attached in here? The rough endoplasmic reticulum is where most protein synthesis occurs in the cell. Why? Because ribosomes are the one that makes the protein, as we have learnt in here. They make proteins, yes? So the rough endoplasmic reticulum is where most protein synthesis occur because of ribosomes. The function of smooth endoplasmic reticulum is to synthesize lipids in the cell. The smooth endoplasmic reticulum also helps in the detoxification of harmful substances in the cell. Next, moving on to Golgi complex. It is the organelle in the cell that is responsible for sorting and correctly shipping the proteins produced in the endoplasmic reticulum. So once the proteins are being produced by the rough endoplasmic reticulum, they'll have to be sent to specific areas where there is need of protein. Yes? So the job to send to that specific area is the job of the Golgi complex. Just like our postal packages, which should have a correct shipping address, the proteins produced in endoplasmic reticulum should be correctly sent to their respective addresses. It's a very important step in protein synthesis, yes? If the Golgi complex makes a mistake in shipping the protein to the right address, certain function in the cells may stop. This organelle was named after an Italian physician, Camillo Golgi. He was the first person to describe this organelle in the cell. Okay. Vesicles. This term literally means small vessel. This organelles help store and transport the proteins that were produced to the different parts of the body. The vesicles are transport and delivery vehicle like our mail and Federal Express trucks. Some vesicles deliver materials to the part of the cell and other transport materials outside the cell in the process called exocytosis. Moving on to vacuole. Remember we learned in the difference and similarities of animals and plant cells that vacuoles are only present in plant cells? If you remember that, let's go in detail about vacuole. Plant cells have what look like a very large empty space in the middle. This space is called the vacuole. The vacuole contains large amount of water and stores other important materials such as sugars, ions, and pigments. Peroxisomes and lysosomes. Peroxisomes, these collect and safely break down chemicals that are toxic to the cells. So they help the cell by making it non-toxic. What are lysosomes? They are created by the Golgi apparatus. These help break down larger molecules into smaller pieces that the cell can use. Next, mitochondria. This is one of the most important organelle. Yes, this is the cell's powerhouse. This organelle packages the energy from the food you eat into an ATP molecule. Every type of cell has a different amount of mitochondria. There are more mitochondria in cells that have to perform a lot of work. For example, your leg muscle cells, heart muscle cells, all these perform work almost the entire day, yes? Other cells need less energy to do their work and hence have less mitochondria. Next, moving on to the chloroplast. And this was also another organelle that's only present in the plant cells, yes? This is the organelle in which photosynthesis takes place. In this organelle, the light energy of the sun is converted into chemical energy. Chloroplasts are found only in plant cells, not in animal cells. The chemical energy that is produced by chloroplasts is finally used to make carbohydrates like starch that get stored in the plants. Chloroplasts contain tiny pigments called chlorophylls. Chlorophylls are responsible for trapping the light energy from the sun. Now that we have learned all the different organelles, let's compare the characteristics of animal cells and plant cells. So how are they classified? Both the animal cells and plant cells are eukaryotic cells. Yes, they are, which means they are multicellular. Yes. How about the cell size? The animal cells are smaller in size, whereas the plant cells are usually larger in size. We already saw that. How about the cell shape? The animal cells have irregular shape, and the plant cells have fixed rectangular shape. Movement. Cells can move around by changing its shape. They have limited movement. Plasma membrane. Plasma membrane is present and contains cholesterol. What do you think about the plant cell? Will it have cholesterol? Plasma membrane is present but does not contain cholesterol. Cell wall. Can you tell me if the animal cell will have cell wall or will not have cell wall? Yes, they don't have cell wall. The plant cells, they have cell wall. How about the vacuole? Will the animal cell have vacuole? One or small temporary vacuoles, much smaller than the plant cells, very small ones. Plant cells, they have one large permanent central vacuole taking up to 90% of the cell's volume. Amazing, yes? Chloroplast. So do the animal cells have chloroplast? No, they don't. Do the plant cells have chloroplast? Yes, they have to. That's the main organelle for the plant cells because that's where the photosynthesis happens. How about nucleus? I'm sorry, the spelling is spelled wrong in here. Nucleus present in the center of the cell. In the plant cells, nucleus present along the periphery of the cell. Centrioles present in all animal cells. Only present in lower plant cells, plant forms. Example, Chlamydomonas. Golgi apparatus. Have a single highly complex Golgi. Have several simpler Golgi. Mitochondria. They are present in animal cells and also present in plant cells. Endoplasmic reticulum. Present. Lysosomes. Present. May be present. Vacuoles also function as a degrading site.